Namaste to all students. Khusho Bitia, myself Jeevan, Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about cyclin and cyclin dependent kinase and its role. Basically, cyclin and cyclin dependent kinase are very much important enzymes from cell division process, and this session is related to cell biology course of program BTech FBT and BSc FST second sem. Cyclin and cyclin dependent kinase, both enzymes are required for successful process of cell division. If we talk about the most important core cycle regulators, so proteins which are called cyclins and enzymes called cyclin dependent kinase create an enzyme complex which is known as APC oblique C. First of all, we have to know about cyclin and cyclin dependent kinase. So cyclins are among the most important core cell cycle regulators. Cyclins are a group of related proteins, and there are four basic types found in human and most other eukaryotes. For example, G1 cyclin, G1 oblique S cyclin, S cyclin, and M cyclin. As the name suggests, each cyclin is associated with a particular phase of cell cycle, transition, or set of phases in the cell cycle, help drive the event of that phase or period. For instance, M cyclin promote the event of M phase, such as nuclear envelope breakdown and chromosome condensation process. The level of different cyclins vary considerably according to the cell cycle phase. As you can simply understand in the diagram which I am here going to show, and a typical cyclin is present at low level for most of the cycle, but increases strongly at the stage where it is needed. M cyclin, for example, peak dramatically at the transition from G2 to M phase. On the other hand, G1 cyclin are unusual in that they are needed for much of the cell cycle. Here you can understand all the peaks of cell cycle when they are required. So you can simply understand this is concentration, and these are the phases: G1 phase, S phase, G2 phase, and M phase. G1 cyclin is start to get the peak from G1 phase, and G1 oblique S cyclin will be highest in the S phase. S cyclin will be highest in the S phase, and M cyclin will be highest in the M phase. Now come to the second important factor, which is cyclin dependent kinase. In order to drive the cell cycle forward, a cyclin must activate or inactivate many target protein inside of the cell. Cyclin derive the event of cell cycle by partnering with a family of enzyme called the cyclin dependent kinase CDK. A lone CDK is inactive, but the binding of a cyclin activates it, making it a functional enzyme and allowing it to modify target proteins. How does it work? Cyclin-dependent kinase are kinase enzyme that phosphorylate, means attach phosphate group to specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. When a cyclin attaches to a CDK, it has two important effects. It activates the CDK as a kinase enzyme, but it also directs the cyclin-dependent kinase to a specific set of target proteins, once appropriate to the cell cycle period controlled by the cyclin. For instance, G1 oblique S cyclin send CDK cyclin dependent kinase to S phase targets. Example is promoting DNA replication, while M cyclin sends cyclin dependent kinase to M phase targets, for example, making the nuclear membrane breakdown. You can understand about the concept of cyclin and cyclin dependent kinase enzyme. So here you can simply understand two possibilities. If there is no cyclin, so cyclin dependent kinase is inactivated. So it can't go for the target 1, target 2 or target 3. So simply the S phase factors are completely off. When cyclin is present, means G1 oblique S cyclin. Here I'm just giving an example. G1 and S cyclin, if it is present, so it will bind to the CDK, which make it activated. Now this CDK will phosphorylate their respective targets. Here you can simply see this target 1 is phosphorylated, 2 is also phosphorylated, and third one is also phosphorylated. It is completely just because of CDK activation by G1S cyclin. After the phosphorylation of target 1, 2, 3, S phase begins, means DNA replication enzymes are now activated for the process of DNA replication. Now come to a factor which is maturation promoting factor MPF, a famous example of how cyclin and CDK work together to control cell cycle transitions is that of maturation promoting factor MPF. The name dates back to the 1970s when researchers found that cells in M phase contained an unknown factor that could force frog egg cells stuck in G2 phase to enter M phase. This mystery molecule called MPF was discovered in 1980s to be a CDK bound to its M cycling partner. You can understand this concept by this diagram. For example, if there is M cycle present, so it will activate its respective cyclin dependent kinase. When CDK is activated, so it will phosphorylate its respective targets. After getting the phosphorylation of targets, M phase begins, means mitosis division starts, spindle fiber forms, chromosome condensation happen, and nuclear membrane breaks down. So example, if we are talking about the role of M cyclin, so nuclear membrane breakdown is the result of M cyclin activation of CDK. When nuclear membrane start to disappear and chromosome start to condense, so cell go for the mitosis process. Here you can simply see the difference after the activation of M cyclin CDK. Here this is complete nuclear membrane, but when M cyclin bind to the CDK, it activates, and then this CDK phosphorylates its targets. When targets are phosphorylated, they go for the signals, and now membrane start to break down. So this change start to happen during the M phase after the activation of M cyclin and CDK. Next complex is the anaphase chromatic complex, or you can call it cyclosome, APC/C. In addition to driving the event of M phase, MPF also triggers its own destruction by activating the anaphase promoting complex oblique cyclosome. APC/C a protein complex that causes M cyclin to be destroyed starting in anaphase. The destruction of M cyclin pushes the cell out of mitosis, allowing the new daughter cell to enter G1 phase. The APC/C also causes destruction of the proteins that hold the sister chromatids together, allowing them to separate in anaphase and move to opposite poles of the cell. You can understand more about this anaphase promoting complex by this diagram. This is anaphase promoting complex oblique cyclosome. It adds ubiquitin to securin. This securin is generally bind to the separase enzyme and which make it inactive. So when anaphase promoting complex is present or is activated, so it will add ubiquitin to the securin protein. On the other hand, if we are talking about these two sister chromatids, they are bind to each other with cohesin. When securin is added with the ubiquitin, so it will release the separase enzyme, and this securin will go for the proteasome, which is recycle bin. It will degrade this securin protein. Now separase enzyme is completely free and it is active in form. So this activated separase now go and break down the cohesin, means they chopped up the cohesin, by which these two sister chromatids are able to separate it from each other. So that's how anaphase promoting complex or cyclosome do work. They basically degrade the securin by adding ubiquitin to it. After adding the ubiquitin, this securin will be degraded or broke down by the proteasome. After the degradation of securin, this separase will be free and will be activated. This activated separase now degrade or chopped off the cohesin, which is responsible for the binding of sister chromatids of chromosome. So when it is chopped up, so these two sister chromatids are separated from each other. That's how anaphase promoting complex do its work. So these are some important points related to the cyclin and cyclin dependent kinase. So these two enzymes are very much important for the successful function of cell cycle. Again, if I repeat, there are various cyclin like G1 cyclin, G1/S cyclin, S cyclin, and M cyclin. These are four major cyclins which are responsible for the successful cell cycle. As we can understand by the name of these cyclin, G1 cyclin is useful for the G1 phase, G1/S cyclin is responsible for the end of the G1 and starting of the S phase, and S cyclin is useful for the S phase, and M cyclin is responsible for the M phase completion. But this cyclin is not capable to do successful process of cell cycle. It requires a accessory enzyme which is known as cyclin dependent kinase. Along with cyclin dependent kinase, this cyclin do its function and do help in promoting the events of all respective phases, such as nuclear envelope breakdown in M phase and chromosome condensation in M phase happens by the MEM cyclin along with its respective cyclin dependent kinase enzyme. Cyclin dependent kinase enzyme basically phosphorylates its target, and after the phosphorylation process of target of a cell, these targets, phosphorylate targets, further help to promote the final event of that phase. Some other factors are also important, like anaphase promoting complex, maturation promoting factors. These are very much important for the regulation of cell cycle. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.